Welcome to our community of practices for the Berkeley Superfund program. Today we had on our schedule two speakers, but one of our speakers had to cancel at the last minute because of illness. So we have the pleasure of having the entire meeting dedicated to the interesting work of Professor Andreas Stahl. Professor Stahl is the Ruth Oakey Chair in the Department of Nutritional Science and Toxicology at UC Berkeley. He works on issues related to lipids and fats, and today he's going to tell us something about using organs on a chip to understand endocrine systems and how that might be related to some of our interests here in the Superfund program, particularly as it relates to PFAS. So Andreas, take it away. It's all yours.
Yeah, thank you, David, for that kind introduction. Thanks everyone for joining. So this PFAS program is relatively new to me as well, as I should say. And while I was including PFAS here in the scheme of things where we can stimulate our organ-on-a-chip devices, as a full disclosure, I have not experimented at all, at least knowingly, with PFAS in our system. And this is really, I think, an opportunity for me and the group to talk about forward-looking experiments of how we could potentially leverage this technology, this organ-on-a-chip system, that I'm going to talk about more in the next 14 minutes or so, to look at these persistent chemicals in the environment. But I'll focus right now on what we've done so far, which has mostly been to establish milieux, circulating milieux within these microfluidic devices that are powered by stem cells, particularly human iPSC-derived adipocytes and hepatocytes, to investigate obesity-related disorders as well as aging-related disorders. But I think, again, that they have the potential, in my mind, to expand on the scope of what they can be used for. So with that, let me see that we advance the slide. No. Let's try this. All right. So what are organs on a chip? Sometimes also referred to as microphysiological systems, and why use them with stem cells. The main advantage that we have with these microfluidics, and the picture on the left here is actually one of our chip setups, is that you can engineer in very clearly defined systems for both how the fluidics work in terms of the flow of medium over the cells, so you don't have a static system as you would have in classical tissue culture. You can shape the environment of these cells. You can promote growth in 3 dimensions rather than in 2 dimensions. You can minimize the amount of medium that is needed for your experiments, which becomes critical when you're using rare resources such as human serum, as I'll show you later on. And you can interconnect these systems to allow reproduction of inter-organ communication, which is also very hard to do with classical tissue culture. And then why IPS? So people have used these microphysiological systems with primary human cells as well, particularly hepatocytes. But we firmly believe that IPS is the way forward just because of a higher reproducibility. You have a defined genome here. All of the cell lines that we're using are fully sequenced, and they can reflect human diversity in a controlled way, but they are also amenable to genetic editing, and that allows us to investigate, for example, drug genome interactions, introducing higher risk factors. For example, PNPLA3 is the poster child of a high risk factor for fatty liver disease. So all of these can be engineered. So I think it's a very powerful combination. What we are modeling here mostly are 2 tissues, adipose tissue, fat, and hepatocytes, the 2 main cell types of fat and liver. And the reason for that is that fat and liver compose the central axis in your body that regulates fasting feeding transitions, and dysregulation in that crosstalk between fat and liver underlies much of what you see in obesity-related disorders, such as type 2 diabetes, fatty liver disease, etc. So we're very interested in that. We spent as a lab quite a bit of time optimizing the differentiation of iPSCs into human adipocytes. That was something that previously had not worked well and yielded cells that were not responsive to stimuli such as insulin, which of course is a key factor in terms of regulating adipocyte biology. So we managed to optimize this, and this here is a snapshot. I hope you guys can see my laser pointer here, of a well. So these are round wells. They are 100 microns high, and that allows cells to form an actual three-dimensional tissue that we support through the introduction of a matrix, an artificial matrix as well, but the cells then start to make their own matrix in the end. They sit underneath a membrane. I didn't have a slide here to show how the device itself looks like. I'm happy to explain that in more detail if you guys have questions about that. One of the things for the fat connoisseurs here to point out is that normal adipocytes in tissue culture have many, many lipid droplets, many small lipid droplets, while if you take a section of fat from a human, these cells only have a single lipid droplet. And what we're actually getting here in this three-dimensional tissue culture are also mostly cells that have unilocular lipid droplets, which is quantified over here, which is great, which means that we are creating a more physiological tissue. Now, the important thing is that we can run functional tests with these adipocytes as they sit in the wells, and we're very interested, as I mentioned, in obesity. And of course, insulin resistance is the main thing that you want to look at in that context. So we developed 3 assays that we can run within the device with the adipocytes that are readouts of insulin sensitivity that includes the ability to stimulate glucose uptake. So just like skeletal muscle, adipocytes have an insulin-responsive glucose transporter. We can look at fatty acid uptake. There's also an insulin-responsive fatty acid transporter. And importantly, we can also look at the regulation of lipolysis, which is stimulated by beta-adrenergic stimulation. You can see lipolysis going up. And then if we add on top of that stimulation with isoproterenol insulin, that then suppresses back lipolysis. And that's actually one of the important things that goes wrong in the state of obesity, where you have a failure of insulin to suppress lipolysis. So we start out with healthy, good-looking cells, and we then can manipulate the system to see whether we can introduce drugs, compounds, and again, here you could envision that we would introduce something else, like an environmental pollutant as well, and then look at the impact of these drugs and compounds on insulin sensitivity. One poster child here is metformin, a known insulin sensitizer that actually enhances the response of these cells to insulin. But maybe more interesting in this context, we can also look for insulin desensitizers. And you probably are familiar with the fact that many drugs in the clinic right now have unwanted side effects that include the induction of insulin resistance. The poster child for that is normally anti-inflammatories. And indeed, if you look at hydrocortisone, you see here that the entirety of the insulin-stimulated response is wiped out. And this is just overnight treatment of the cells. Potentially scary to some of you, statins are also known to increase the risk of diabetes. Treating the cells with one of the common statins, atorvastatin, also completely wipes out insulin sensitivity. So that was a new finding here. And again, this holds true for all of the different assays that we can run for insulin sensitivity that these compounds either stimulate or inhibit. And you can scale this, as you see on top there. Those are some of the devices here that are multiplexed. We can easily bring this up to 96 wells on one of the glass substrates. We can then make the system a little bit more complex to further mimic what happens in the state of obesity, which is that you not only have an expansion of adipose tissue in the obese state, but you also have inflammation in the adipose tissue. And most of this inflammation, also not all, but most of it is really driven by the ingression of macrophages that are M1 polarized macrophages that are pro-inflammatory. Now, we wanted to recreate this in our system as well. And we wanted to stay isogenic, meaning that these macrophages should come from the same IPS that we use to make the adipocytes. So that's what we ended up doing. We differentiated IPS into macrophages. We can polarize these macrophages either towards an pro-inflammatory M-like phenotype or a tissue repair-like M2 phenotype. And we can then, and that's shown in that little video here on the left of the slide, flow in these differentiated macrophages into the already resident adipocytes. And what you see over here on the right is with the macrophages in blue, the cells are originally on the outside around this cluster of adipocytes, but then within hours, they are starting to ingress into the three-dimensional cluster of adipocytes and start to interact with them, as you would see in normal human tissue, where you always have macrophages clustered around adipocytes as well. So we can establish these co-culture conditions, and we can then ask, what is the consequence of flowing in either pro-inflammatory or anti-inflammatory macrophages? And we had a paper where we showed that pro-inflammatory macrophages drive insulin resistance. And I can point you towards the publication, but I wanted to stay within what I thought was my original time frame. So I won't show you the data, but again, you can look at the effects of drugs on top of the introduction of the pro and anti-inflammatory macrophages. But as a next step, and this is what I do want to share with you, is we can now not just look at adipocytes and inflamed or non-inflamed adipocytes, we can look at the effect of these adipocytes on hepatocytes. As you know, fatty liver disease is one of the complications that come about with obesity. About 1 in 3 Americans suffers from fatty liver disease. And there are currently still no drugs that are approved for the treatment of fatty liver disease. This is actually a disease that's hard to mimic in animal systems because of differences in lipid handling in mice and humans. So we are and often impractical to study in animal models where lipid handling would be similar, for example, in pigs, but pigs are very expensive to work with. So here you have now a system that's still isogenic, where we have hepatocytes derived from one IPS line. From the same IPS line, we derive adipocytes, and we can then also introduce macrophages. We optimize the co-culture conditions, and we can interconnect these devices, as is shown here, to basically recreate this inter-organ communication between the adipocytes and the hepatocytes. And this is shown here again. You have the adipocytes on one chip, the hepatocytes on another. We can set these up as 2 separate devices, so you can initially address them separately and then join them, or we can integrate them into one substrate. These are all PDMS-based designs, and have an integrative, always connected circuitry as well. So both is possible. This is the interconnected device here. And what you then can do is ask interesting questions. For example, is there a flux from fat that is stored in the adipocytes into the hepatocytes? And we can monitor that because everything is on a glass substrate. A microscopy is very easy to do with these organ-on-a-chip systems. And what we have done here on the left, what you see is adipocytes that have been loaded with a green fluorescent fatty acid. And we interconnect these adipocytes with hepatocytes. And what you see is that there's very little transfer of fat from the adipocytes to the hepatocytes. And that's because these are healthy adipocytes that do a good job regulating lipolysis and holding on to the lipids. But when we then introduce macrophages into the adipocytes, and particularly pro-inflammatory macrophages, we create inflammation in the adipocytes, we create insulin resistance in the adipocytes, and that now leads to spilling out of the fat from the adipocytes into the hepatocytes, effectively recreating what you see in obese patients that are developing fatty liver disease. And we can look at gene expression in these systems, and you see that TNF-alpha and IL-6, 2 very well-known pro-inflammatory cytokines are massively upregulated. Cytokines associated with the state of obesity, leptin, are upregulated, while other adipokines that are for insulin sensitivity, that promote insulin sensitivity, adiponectin, are downregulated. So very much matching what you would see in an obese patient. This is on the adipocyte side. We can look at the hepatocytes as well, and we would see upregulation, particularly of genes involved not just in inflammation, but also gluconeogenesis, which of course is what happens in the liver when you develop insulin resistance. The liver fails to shut off the release of glucose into the circulation, and that's exactly what we're seeing in these interconnected devices. Here's a closer look again at the cytokine levels, which we can look at by ELISA as well. Here's a closer look at lipid metabolism and uptake. We can measure things such as fatty acids, glycerol, and LDH. And here measures of insulin sensitivity for the separate devices, again comparing the adipocytes without the inflammatory macrophages and with. This is insulin sensitivity adipocytes. Here's insulin sensitivity as a readout for the hepatocytes, which is the ability to suppress the release of glucose rather than to regulate the uptake of glucose. Normally, insulin suppresses it. When you have inflammation, the hepatocytes lose that ability to regulate it. And again, you can, now that you have a disease system where we now have the inflamed adipose tissue with the hepatocytes, ask how are drug responses going to look like in this inflamed system? How can you improve insulin sensitivity? How can you rescue it? Again, you can see this is very guttural here, our lipid transfer assay, which is just one out of several assays that you can get from the system. You see that metformin improves the ability to retain lipids, so does rosiglitazone, which is a PPAR gamma agonist. But trying to dampen the inflammation with dexamethasone does not work. So there are important and informative things that you can get out of this. We, for example, tested also the response to semaglutide, everyone's favorite drug response, everyone's favorite drug at the moment. And there are actually effects of these of semaglutide and GLP-1 receptor agonists outside of the brain, and we can show that indeed this too improves the situation here and actually improves the communication of adipocytes with the macrophage. This, and I know I went a little bit over time, but I hope I'm still within the 2 time slots that I ended up occupying, was a really brief presentation of what we're able to do with these microfluidics. I do want to give a shout out to Qing Fang, who pioneered, she's right here in the center, who pioneered the differentiation of these adipocytes from iPS, and Lin Chi, who did many of the experiments and produced the data that I showed you on the aging on a chip phenotypes. So with that, I hope we still have plenty of time for your question. Thank you.
I don't think we have plenty of time, but we have a little bit of time.
All right.
So I guess I'll throw it open to people on the meeting who might want to ask a question. I'm looking to you biologists especially. I think it's a Monday morning shy thing, Andreas. So let me start by just kind of pushing you a little bit on this question of PFAS. So, I think if I understand correctly, a lot of the impacts of the known impacts of PFAS are on the lipid system and similar types of endocrine pathways. What are your thoughts for the likelihood? Like, have you seen people publishing work on lab on a chip with PFAS that gives you hope that you might be able to bring those chemicals into your system?
I have not seen PFAS with a microfluidic system, but again, we are really one of the few labs that have pioneered the use of iPS-derived adipocytes. There could be some information out there using liver organoids, particularly as it comes to catabolism of drugs. Those have been widely used. And I believe there's some literature out on it. I'm not claiming to be the world's experts on PFAS, so take it with a grain of salt. But Right now, I don't think that there is much literature on that. Could it be used for it? I'm pretty positive it could. I think one could easily envisioning introducing PFOS directly into the system, either with the adipocytes and hepatocytes separately or in the intercirculating medium. That could mimic both the sequestration into adipocytes, as these are hydrophobic compounds, or the catabolism in the hepatocytes. And at the same time, we should be able to look at the biological effects in terms of impacting viability, but also much more finely grained in terms of impacting insulin sensitivity, for example. So I think that that's one possible way with direct introduction of the compounds. Another possibility would be to have serum from people that had high and low exposure, which would include then, of course, additional factors that may change between these different people. So we may have to pool it, but also could be a possible additional approach.
Great. Other questions? Let's see, hands raised.
I can ask the question. Hi, Andreas.
Yeah.
Hey, it was good seeing you.
Nice seeing you. I guess it's similar in lines with that. A lot of the standards that were set were based on liver toxicity, right? So like lesions. And so we were thinking about in your system, I know there's not many cells. So just trying to figure out how you could potentially recapitulate some of the endpoints that were assessed by the EPA to kind of see if that's something we could use as a model in your system.
So the number of cells in these wells is actually very high because they are packed very densely. But in any case, well, we can look at viability easily, right? We've done this. In fact, one of the readouts that I went way too fast for you guys to see was viability. We can look at ALT, AST. All of those are really easy to actually do. So I think if there is something that would damage or kill the cells, and we've actually tried this now with some drugs where I briefly mentioned that we're interested in sexual dimorphisms as well. As you know, the liver is incredibly sexually dimorphic. So we introduced drugs that affect female livers more than male livers. And we have no problems getting titration curves for toxicity with the system, just using a simple live dead kit.
Thank you. That's helpful.
Great. Cleona, do we need to end because it's 12:30, or is there time for more discussion?
If people have more questions, we could stay on a little.
I'm here if you guys have questions, and if
Yeah.
you have to run.
And Andreas, unfortunately, I'm going to have to go because I have another meeting, but I'll leave
Okay.
it to others to ask the questions. And Cleona, maybe you could take the question from Junsu, who's next.
Yeah.
All right.
Just
I did.
Wow, this is always a kind of a challenging topic for me. So anyway, I just want to make it straight for me. If I actually followed your lecture, Dr. Story was great lecture, by the way, even though I couldn't understand all of them. Yeah, just one quick question was, as David mentioned because this is the PFS, the group. So I'd like to bring the PFS, the work we have done, kind of correlate with some fatty, the lipid metabolites, all the fatty acid kind of kicked in and showed the positive correlations. So having this said, you kind of addressed several the end product. I would call end product. So PFS got exposure to the PFAS. It kind of enhance, let's say, regulate the fatty acid, several fatty acid. Then based on your earlier slide, you had insulin kicked in. And also, you're talking about some aging phenotypes. Are they related, or they are kind of you address as a separate outcomes?
Yeah, so the aging phenotypes are related to the insulin resistant phenotypes. which is what you do see in people as we get older. Our ability to respond to insulin decreases, and that includes a poorer performance to regulate lipolysis. So you see there's actually an increase in circulating free fatty acid levels as well with aging. What would be of interest to me, though, Jun-Soo, is what would be readouts for the PFAS field that you guys would feel excited about? Would it be insulin sensitivity? Would it be regulation of lipolysis? Would it be engaging nuclear receptors and looking at the transcriptional response? What would be functional outcomes that would be high on your list that you would like to investigate?
Yeah, frankly speaking, I'm chemist also and the laboratorian. So my main interest in how to analyze them, how to correlate to the next chemicals. That's why the most of our studies stopped at the let's say we measure PFAS in the blood and how they help the regulating down regulating the metabolites, including lipid metabolites. Then the next outcomes is you just display the listed many interesting outcomes. I think that will be we defer to the either the Rosemary or the Michelle toxicologist. So I don't think I'm a good person to answer to your question, but why not? Why not try to see further after the metabolites kicked in and insulin also is stimulated, then what happens to that?
Yeah.
So
Okay, since you're the chemist, and I have another question for you. Since this is a whole group of compounds, as I said, we could look definitely at 10, 20 or so, but if you say you would have to look at 1,000 different ones, that would be problematic. Are there specific PFAS that you would say these are the top 5 most important ones? And are there maybe some subgroups where you say, I really need a comparison of this group A versus that group B?
Yeah, that's a great question. So as a member of a PFAS group, we used to monitor the most persistent one. We call the C8 PFAS compound. For example, PFOS, the octanoid, the sulfonate, also the octanoic octanoic acid. So those two are the kind of representative of our legacy PFAS. But after then, they've been kind of volunteering phased out or regulated. After then, the new and the replacement PFAS coming out, including not only fluorotellular alcohol type, also the recently, that's why I was interested in what Michelle found out in her toxicological test. We've been very interested in the short chain PFS too. The 4 carbon and 3 carbon, particularly. I don't know when this here, yeah, the whole group the recently measured huge amount of the trifluoroacetic acid, so very short carbon, but they observed that high detection of TFA in their recent the campaign of drinking water monitoring for PFAS. So if we want to also, I suggested before I suggested I believe Michelle too. The kind of looking at some ether group, the supposed to have a very kind of easy to break down in the body, so have a short half-life, but I still show the similar toxicity as the most persistent compound I just listed, the PFOS. There is kind of it's called the Gen X or Aduna, also the F53B, I think that's what the name of the same similar type. A similar type has one has a ether and the chlorine there. So I think if we want to separate some toxicological test, I'm personally interested in the legacy and the new replacement and the kind of short chain.
Yeah, and I would imagine that a C8 versus, something that's more on an acetone or acetate backbone would have a very different distribution between the adipocytes and hepatocytes.
Thank you for asking. I think
Yeah,
try to
no,
myself.
look, I want to learn
Yeah,
from you guys here too.
yeah. No. This is exactly what this meeting is for. Yeah, great lecture.
Thank you. We've gone over by 10 minutes. Does anyone have any more questions?
No.
No, it doesn't look like it. Thank you, Andreas. Everyone, our next meeting on May 5th will be immunological effects of PFAS. So I'll send an update and a reminder, and we'll share Andreas's video as well. Thank you so much.
Thank you
Thank you
Bye,
very much.
everyone.